Hi, I'm Meredith Blackwell, and this is Professor Tom Bruns from Berkeley. And I have known Professor Bruns since he was a very young child. He was just a baby mycologist. He was at the University of Minnesota, and I was helping somebody arrange a symposium. And it turns out we heard about him, and he was working with flies and boletes and hatching flies out of boletes because they eat up boletes. And what happened was we were told he was finishing up his PhD. And it turned out he was still a master's student, but still it was a great paper. So, Tom, tell us about your career. I know that mycology saved your life, correct?
I suppose it did, yeah.
You wrote that once, do you remember?
No.
Tom once wrote an essay, Why I Became a Mycologist. But anyway, where were you born?
I was born in Illinois,
Okay.
outside Chicago. Grew up in the suburbs when they were being built. And so there were lots of prairies around, and I was an avid insect collector for many years. And then I went off to college not knowing what I wanted to do. And I stumbled into a mycology course. I had to take, I was a biochem major at the time, and I had to take something in the life sciences, and so I thought I'd take something with a field trip associated with it, and I took a mycology course from D.P. Rogers at the University of Illinois. And I was really excited by it after that, and I still didn't know what I wanted to do, but I did know that I didn't want to be in the lab my whole life, which is ironic since I've been in the lab my whole life now. But
Well, you have a mix.
I have a mix. I certainly get out and do a lot of field work, so it worked out really well for me. But I didn't. I think, like most people, I didn't really even think of mycology as a possibility for a career. I mean, how many? I don't know, career day at wherever you do it probably doesn't have a mycologist, right? So I didn't really know that was possible. And after that mycology course, or actually even before it, I was starting to collect mushrooms and look at them, but at the time, quite difficult to learn that on your own. But I struggled along, and I learned a few things. And then at Illinois, I didn't finish. I just quit after 3 years because lots of things were going on in my life, and took some time off. Moved up to Minnesota, established residency there, and after 2 years, then I could go to school and finish. And my main reason for doing that was that I wanted to finish in forestry because I thought forestry would be that nice compromise that would allow me to be a scientist of some sort, but still get out into nature, which is what I wanted to do. And then I finished up there in forestry.
Oh, you actually got a degree?
That's my first degree, is forestry, forest science. And probably before I did finish there, I know before I finished there, I realized that forestry was not what I wanted to do. But by that time, I just wanted to get something completed because I think prior to that I'd changed majors maybe every 6 months or so on. And so I finished in forestry, worked for the Forest Service for a very short time on one of those 180-day appointments doing aerial photo interp, and continued to collect mushrooms and got more interested in mushrooms, and came up with the idea of the bolete project, right?
So when did you start early noticing the insects?
Yeah. So anyone who collects mushrooms to eat notices the insects because they're better at finding them than we are, and get their first grab at it. So I got interested in the patterning of their feeding and noticed that they like certain areas and they like certain mushrooms, and I thought there was probably a project there. So I went back to the University of Minnesota, and I talked to 2 mycologists there about this thing I wanted to do. And the first one I talked to told me it was a can of worms, which was probably true. And he had a different project that he wanted me to do, which involved taxonomy of a genus.
And you're not saying who he is. That's okay.
Yeah, I'll protect him, because it didn't turn out well. I think his idea was, you never would have heard from me again if I had done that taxonomic project.
We're glad you didn't then.
Yeah.
And the other guy.
And the other guy was David McLaughlin.
Yeah, and he just walked by. He's at this meeting
And David
too.
McLaughlin said, "That sounds like a great idea. Why don't you apply for graduate school? Come here and do it." And so I did. David, of course, knew a lot about the boletes. He didn't know anything about insects, which was fine. He gave me space to work and hooked me up with teaching assistantships to teach my way through, and I had a great time. I wanted to do a master's, I think, because I still wasn't sure. To me, I didn't ever see myself as being sort of academically bound. I didn't come from a family that did that sort of thing. My father was a florist. My mom was a receptionist. I think I always wanted to be some kind of a scientist, but I didn't know what that meant. And so it was a master's degree was a really good thing for me because I could get a taste of it and see whether I liked it. And in the end, I loved it. I poured myself into it. Found that time flew when I worked on it. Found that you could become an expert in something in just a couple years, especially if it was obscure enough. And so I had a lot of fun with that. And then I applied for grad school at Michigan and went there to work with Bob Schaefer, who was expert in mushroom taxonomy, with the intent of probably doing something molecular. And I was going to work with Wes Brown, who was he was Nobel laureate. No, this is a different one. This was a chemist. Never mind. Yeah, Wes Brown did. He'd worked with Alan Wilson before that and did mitochondrial DNA in animals.
That's right.
And when I talked to him, when I was thinking about going to graduate school, I talked to him and I said, I'd like to do some molecular systematics of fungi. He said, "Well, it sounds like a great idea. Why don't you come here?" And then once I got there, he said, "Well, my lab is for animals. If you want to work on animals, that's good. But if you want to work on fungi, I can't really help you." So I was kind of left in the lurch there with Wes. And so I started continuing on the bolete stuff because that was something I knew, and I thought, okay, I can spin something out of that. But then talking to other people, I ran into Mike Hudspeth, who was a postdoc with Larry Grossman, and he was mapping mitochondrial genomes of oomycetes. And I told him I was interested in molecular systematics with Suillus, and he didn't know anything about Suillus, but he said, "Well, come on up. We'll extract the DNA and map it up and see what we can do." And he was just, can-do attitude.
Yeah.
Do you remember Mike?
No, I don't.
Mike was a character and a half. He, this was still when you could smoke in the buildings, and he always had the cigarette dangling out of his mouth, and he'd wear a leather jacket and rode in on a Harley with his girlfriend at the time, who also worked in the lab, Debbie Chimard. And he was a real character. So at any rate, he taught me how to do it. Taught me how to hold he was really, in many ways, he was my first advisor. Okay. And so those first few genomes that I mapped were all done with his help. And if he hadn't done it, it wouldn't have happened. And then the person he was working with, Larry Grossman, didn't get tenure. So once again, I was kind of in a lurch as to where to work. At that time, they just hired Jeff Palmer, a plant systematist. And so I went and talked with Jeff, and at the time he had an empty lab, but a big one. So he said, "Yeah, sure, come on in." And
that worked well.
It worked well, and he was he had a very different style than Mike, and was much more into efficiency. Like, I went from, it took me maybe 3 years to map my first 3 genomes. And when I got up there, we mapped out like maybe 15 more in about a year. he had the method,
And then you gave a paper, or did you do one earlier? A paper at a meeting?
Yeah. So I'm trying to think.
John Taylor was talking about
Fort Collins.
Yeah.
So that was the first paper I gave on molecular stuff, and it was right near the end of my work, or maybe it was more like the middle of it, I guess. And I went and gave the first talk there, and I was sure it was going to be the only one, on mapping mitochondrial genome. And I got there and met
John.
Met John, and he was doing the same thing. And I think at the time, we clicked right away. And I said, why? I think what I said was, why don't I write a grant and come to your lab and continue this? And he said, sounds like a great idea. And I did, and I got the grant and took it off. It must have been near the end.
Yeah.
It must have been near the end, because I think I had like a year left to go or something. But yeah, that's how it started off. So there, I didn't hear John's interview, but I hear he mentioned that there was a lot of luck involved with his career.
Yes.
I would have to say exactly the same thing. That things clicked in very idiosyncratic, unexpected ways, and it worked out well.
Yeah. I think many mycologists have a lot of luck, because we don't start out to be mycologists usually.
Yeah.
You almost did, but except for the forest ranger days. But I was going to be a architect, and then an English major, and then a history major, and then political science, and finally went deep sea fishing and wanted to be a marine biologist. But I ended up pure luck working with Dr. Helix, because I didn't get along with the fish guy at Texas. So it's all a matter of luck. So I ran into you at Berkeley, but you got there, and I think John or you told the story about how John hired you and then left, went to Australia?
Yeah, so I got the grant. The grant was on the boletes, right? And so I was going to do molecular systematics of the boletes with this mitochondrial mapping thing. But in the meantime, I learned to clone, and we started to sequence. And so I had a few of the first sequences. I think when I went to Regensburg was my first IMC meeting.
Oh, okay.
And I had sequence data then on some of the boletes, and I thought, cool, But very slow process, of course, and then PCR came along. And the way it came along in his lab, did he tell this story?
Not completely.
So it was pretty funny that I was working there one day, and this guy comes in who I'd never met before, and nice guy, and he introduced himself as Tom White. And he said he had this method called PCR, and he had an interest in fungal systematics, and he wondered if he could just work in the lab for a bit. He was a friend of John's. He knew John was out of town. And of course, I'd heard of PCR at the time, but we didn't have it working in the lab. There were no thermocyclers around. It was hard to actually do it. And so as soon as he said that, it's like, yeah, you can have my bench if you want to do this. It's like, come in here. So we hit it off well, and he helped us enormously in that he gave us access to primers so we could design some and try them, and gave us access to thermocyclers that weren't easily available at the time. And he actually was not in the lab very much because he was a busy guy, even though he was taking a break from CETUS. But Steve Lee was in the lab, so Steve and I were trying new primers and cranking it out. And meantime, John was off in Australia at the time, and he'd sent a fax to me because there was no email. And there's only one fax machine on campus at the time. So I had to go across campus to get this fax. I thought, yeah, I'll get it tomorrow. And then tomorrow ended up like days later. I finally got over there, and what the fax said was this guy named Tom White was going to come, and I should see what I could do to accommodate him. So I just wrote back, already done.
And lucky that was. Another lucky streak.
That was lucky.
Yeah. So how many, you ended up with the paper in a book as a book chapter on the primers.
Yeah.
But I'd already been in the lab where I met you again, and Stephen, and John was back from Australia by then. And so it was really nice, and everybody shared with me. It was very difficult in those days because I didn't know how to use a pipetter.
Now, the thing I remember about you being in the lab was that one sort of gel tragedy where
oh my gosh.
Where it broke or it fell. I remember we had to take down these acrylamide gels and fix them in
acetic acid.
Acetic acid, and it got all crumpled up, and you know it's
was
flaming hot radioactive, and we're trying to get this thing in. And you called me in, and it's like, okay, you know, we just can shake this thing out, and it'll straighten out. And we got it back out, and we've got it on the blotter paper and dried it.
It was a gorgeous pattern.
It ended up being
beautiful.
Fine.
And I remember that we'd loaded them two different ways. Two people had loaded the gel, so we'd done it backwards, and so it made a nice kind of Rorschach diagram with the lanes. But anyway, that was fun.
But the other thing I remember about that was, I think when you first came into the lab, I you know, I've always been sort of blunt and tactless. Have you noticed that? The quintessential
You're lovable.
Scientist of like, this is bullshit kind of approach. Mm-hmm. And I think I asked you, you know, are you really serious about this? Are you going to pick this up like at this stage in your career?
I'll tell you exactly where it was. You and Barb had taken me to the botanical garden, and we're walking in the Berkeley Botanical Garden, and you asked me, do you just want to learn how this is done, or are you actually going to do it? And that was where it was. I remember.
Yeah. Mm-hmm. Yeah. And you said, no, I'm going to do it. You know, and you did.
I did.
You did.
I did. You probably didn't believe me.
So yeah.
Did it by the hardest.
Well, I think, you know, a lot of people take sabbaticals where it's more of a vacation almost. They go off and they have time to write stuff up and they learn a few things, but it doesn't really. And you were on, in that point in your career, I thought you were, you know, on in your career ways, and maybe I didn't know you'd go for another 30 years after that.
I got started late. I had to keep on. Yeah, it was just great, gave me new life. And I had a project in mind, and that's why I pursued it. We just discovered Pixie Opera, and David Malik and I we'd concluded separately that it was a relative of the Lobulbiniales, and this is how the lobules came about. And so that's what I wanted to show.
You know, this makes me think just of the early days of molecular systematics. It was like an Easter egg hunt.
Yeah.
Like everything you did, you'd find something unexpected and new. It was just like a door opened, and there were all these possibilities, and more than you could even anticipate. You know, and in fact, I mean, this is actually what got me my job, right? Was the first thing I did was look at rhizopogon and its relationship to Stiwillus, and I have to credit Bob Fogel for that. He was the one. He was the one who started that when I was in Michigan. I was just mapping Stiwillus. I didn't think rhizopogon was related, but I didn't know it much. And someone had published something else on where it was related based on what they thought, and I thought, oh, that must be true. So I didn't think it was related to Stiwillus, but Bob did. And I remember at the time I was really busy growing these things up and stuff. I said, well, look, if you have the time to get this into these, we grew them in these big carboids at the time. If you have the time to grow this up, I'll extract it, map the genome, and we'll find out. And so this was back at Michigan, and I did that, and on the first gel, it was so obvious that it was related to Stiwillus closely that I wrote him right away. I said, Bob, this is a science paper. This thing is
but
so.
It was Nature, wasn't
it? It only ended up in Nature. Yeah, science wouldn't look at it. Science wouldn't look at it. I've never written anything that science would ever look at. You know, I've submitted a few things, but they always like, thanks, but no thanks. But
Well, I remembered nature because you were talking about the next year a study was done by a high school student, and it won #1 in, was it the GE or the Westinghouse?
Westinghouse, yeah, this was...
Yeah, so the first
this was
one
Liz
year,
Pine, yeah.
1 year it's a nature paper, and the next year it's a high school project. I
yeah,
remember you
yeah,
saying
yeah.
that. You were going around
Yeah,
the
yeah. Well, and you look at the, and so we mapped it all up, and the mapping, if you remember, was crude. I mean, you know, you'd get like one character that would support a branch or something, so the trees sucked. But you know, they'd capture some of it.
Yeah.
So what it captured accurately was that it was closely related to Suillus. From the mapping, it looked like it was within Suillus and came out. Fortunately, the first version of that got rejected. They said, well, it's interesting, but you know, get some better data. And so when I got to Berkeley, first thing that happened there was sequencing of that material. And then it was clear that, okay, this thing's really related. But that first sequence paper in Nature, I think had 9-10 sequences in it.
Wow.
That was it, you know, and there was little bitty sequences.
Yeah.
But it did tell the story, and it turned out to be correct and upheld with all the rest of it. But
neat.
Yeah.
Yeah. So I was there for your job interview.
I had forgotten that.
The postdoc left the lab with his briefcase all dressed up and went and had a job interview in part of the building.
Yeah, it was actually the next building over.
Next building.
It was across, yeah. So the other side of that job interview thing was that it was in plant pathology, right? I was working in botany as a postdoc. It was in plant pathology, but you know how it is in academia. Usually if you're a postdoc at the school, you have like zero chance. And so this was the position was a fungal geneticist, which I didn't really do. You know, this was, I guess, molecular.
DNA
it was
stuff.
Molecular genetics,
Yeah,
so we could kind of pass it off
trend.
as genetics. But I thought, this is never going to fly. They're not going to buy this. But and I wasn't even going to apply. I just thought, well, you know, I'm wasting my time, and I better invest the time in writing a paper or doing another gel or whatever. And Steve Lee said, Bruns, you're nuts. You should apply for this. You're right here. Apply. So he shamed me into it. And I think I got my application on the very last day that the application should be in. But before my application went in, I was already signed up to give the seminar because it was just a departmental seminar, and I was, you know, a cheap addition, right? So the word went around that I might be a candidate and that here was a seminar, but it was kind of like an unofficial job, you know, seminar. And so I went to give it. And then the story.
And then I remember afterwards, I was walking out of the building, and there you were standing on the corner with your briefcase, waiting for Barb to come pick you up after your job interview. I thought that was kind of funny. Anyway, so it didn't.
I thought you were going to tell a slide story.
Oh no, tell a slide story.
So the other good thing, of course, was that, or bad at the time, it seemed like, was that talks were given with slides, with real Kodachrome slides, you know, not
2x2s.
You had to make them in advance. Check them out. Always a problem for me since I can't spell. I'm always got a misspelling, and then you get it back and go, got to reshoot the whole roll and get that one. So I had my slides. I go to give the talk. A slide sticks. John Taylor gets up to fix it. Proceeds to dump all my slides on the floor. This is like 10 minutes into my seminar that was ultimately a job talk seminar.
I wasn't there. So.
Okay.
For some reason, I didn't go to your seminar. I would have thought I would have, but I didn't.
But at any rate, because I didn't take this that seriously, and I didn't think I had a chance, I just started laughing. I thought it was really funny, and said, "Well, I'll just go ahead and give this without the slides. And so I went to a chalkboard and I started to draw out, okay, here's the way it looks. And after about 5 minutes of that, it was pretty clear it wasn't going to come across well. And they said, why don't we take a break? You can grab a few slides and put this together. And that was the best thing because I had way too many slides. When I had the 5 minutes to put it together, I could just pick through the 10 I needed, put those in, produce a coherent story, and people remembered it forever because it was this, they said it was the bravest seminar they'd heard. So after that, I became a job candidate, and then a month or two later, they interviewed me one-to-one, like they do for job candidates.
Okay. That may have been the time I saw you with your briefcase and your suit.
Maybe. And it was turned out I was one of 7 candidates that got interviewed, and they were full professors. It was like a wide open search, but I got it. And I was just shocked. And probably ill prepared too.
It was probably the laugh that
it was probably ill prepared too, because I have to say it took me a long time to sort of figure out what the job was about. I was not from that academic tradition, and I didn't really.
Yeah. And then they changed colleges.
Oh yeah, so my department went under, and I got into another department, and then they start a new one. I move over there. So by the time I went up for tenure, I was in my 3rd department.
Was that the one John was in?
By that time it was the one John was in, and obviously it went well. I got things out, and some of my papers had big impact and whatnot. So it went well, and I did figure out the job eventually, and.
Well, I think it's all gone very well because last night you were given the Distinguished Mycologist Award of the Mycological Society. Were you surprised? No, they told you.
They told me. They told me. And, I've seen that go by many times and seen people like yourself get it, and John Taylor and many others. And it's certainly an award I always coveted. I had hoped that someday I would get that award because to me the MSA is just an amazing society. It's really my tribe that talks the same language and has interest in the same sorts of things. So it meant a lot to me.
Yeah.
It certainly meant a lot to me.
Yeah, and you've done well in the society because you've won other awards too. You won Alexopoulos, didn't you?
Yeah.
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 Yeah, it's a good society. So, are we forgetting anything that you want
to? Compress 30 years into the next. I think the last thing I would say is I've just had tremendous students over the years. That many of the people who just walked into my life at Berkeley were wonderful people to work with. Smart, motivated, nice people that I learned a ton from. You always think of professors mentoring students, which is true to some extent, but you learn a lot from your students, don't you? Every one you learn something from, because they all have different interests, and I think because as a student I was always able to explore my own interest. I assumed that students would want that same freedom, and I think it's not always true. Like some of them definitely want it laid out for them,
right? But the really good ones
know what they want to do, or know kind of what they want to do, and so it's not that hard to go, well, here's a bunch of options on where we could go with this, and then they just take off. And many times you'll point them in one direction, and they end up over there, and it's a better direction. Like they saw the right way to go on that. So really, it's just been a pleasure working with students and postdocs in my lab. And then when Louise Glass came to Berkeley, John Taylor and I merged our labs, so that we had one big lab space and shared it, and all of our students intermixed. That was the best thing ever.
With her too?
No,
she was separate.
She actually took my old lab space.
Okay.
And in order to get her here, we needed lab space, and so joining labs was a means to an end, but
it had other benefits.
But it had amazing benefits that I think neither John nor I really anticipated. We got along well, and our students got along well, so we thought, okay, it won't be a problem. But it was not just not a problem, it was wonderful, because it made the sort of critical mass of people thinking about similar problems much higher. So you have these informal interactions every day, and it was great.
And then just one other thing I wanted to bring up is how you started this project, the North American Microflora. It was your idea. And how's that progressing?
Okay, so North American Microflora came about because of FESAN. Do you remember FESAN,
right.
The Fungal Environmental Sampling and Informatics Network?
I'm glad you remember.
which was one of these NSF network grants that allowed us. I forgot who coined the term "meet and eat" grants, but that's what they are. They're grants that allow people to get together and talk about things. And so, mostly what we did was talk about methodology and databases and things like that related to the high throughput sequence revolution and how we were going to coordinate that. And I think it had a huge impact doing that. But because I think all the people involved at the leadership level of FESAN, Karen Hughes and Betsy Arnold, were record cheapskates. We came in under budget and we had leftover money. And so we had enough money for another meeting, and
that was at Yale.
And that was at Yale, and I thought, let's do a Microflora meeting, and we'll just get all these people, both the amateurs, which is a term that doesn't really cover what they do,
right?
And the professionals together that are interested in macrofungi, and maybe we can get a North American Microflora going. And so we organized that meeting and got a bunch of people together, and it's really taken off with other people now that have picked up the mantle. But I started that snowball rolling down the hill, and it's now picking up snow. And
did I tell you that I even have a project?
Yeah,
for the Congaree National Park.
Yeah. Well, I think part of what's propelled it are all these really nice websites like iNaturalist and Mushroom Observer and so on, where you can post this stuff. And now the next big thing that's happened is we've gotten the sequencing coordinated. These groups have been sequencing on their own, but the data doesn't go anywhere. So now the idea is it's going to all funnel into the right databases and get out there so people can use it.
Yeah.
I really hope I live long enough to see the North American Microflora come to fruition at some level, and we get something approaching a microflora for the whole country. And I have to say that the term microflora is controversial because people go, it's not a flora, it's a mycota.
And we don't have herbaria, we have fungaria.
And being blunt and tactless, I would like to say that that's bullshit.
Yeah.
because the people that we have to convince on this are not each other. We're not preaching to each other. The people we have to convince are people that don't study fungi and might be willing to fund it.
They might know what a herbarium is.
They know what a herbarium is. They know what a flora is. And so calling it a microflora and a herbarium is to communicate to them. We know what we're talking about, and eventually we can do more politically correct terms. But I always think you have to think about your audience that you're trying to reach.
And I'm too old to change now.
Yeah.
Okay, distinguished mycologist.
We're
Thomas
done, are we?
Bruns.
Very good.